A special type of chaperone protein found extensively in neurons in the central nervous system, especially in the limbic areas and brainstem motor structures, Sigma-1 receptors have recently been researched for their ability to act as interorganelle signaling modulators. Sigma-1 receptors are important because they have been implicated to have significant functions in neurological and psychiatric conditions and have recently been noted for their potential role in drug addiction, especially cocaine and alcohol abuse. Over the last 2 decades, there has been a vast amount of research looking into Sigma-1 receptors and their effect on neuronal excitability. Sigma-1 receptors are chaperone proteins residing in the mitochondrion-associated ER membrane, or MAM, that regulate dendritic spine formation and regulate IP3 receptors in order to control calcium signaling between the ER and mitochondrion. As shown in the picture on the right, a Sigma-1 receptor has 2 transmembrane domains that are hydrophobic alpha helices. Sigma-1 receptors also have a third hydrophobic helix that is associated with the ligand binding site for the agonist cocaine. Under some cellular conditions, the Sigma-1 receptors can dissociate from the MAM and translocate to other parts of the cell to act as interorganelle signaling molecules. When bound by specific ligands called agonists, or when the ER is under extreme cellular stress, the Sigma-1 receptor dissociates from the MAM and can translocate to different parts of the cell. Common agonists that cause the Sigma-1 receptor to dissociate from the MAM include psychostimulants such as cocaine and the opioid pentazocine. Ligands called antagonists can bind and inhibit the dissociation and translocation of Sigma-1 receptors. When Sigma-1 receptors translocate, they can bind to a number of different protein structures on the plasma membrane that affect both internal excitation and synaptic excitation. Internal excitation is affected when Sigma-1 receptors bind either directly or indirectly to voltage-gated ion channels. As shown in the diagram, sodium is heavily concentrated extracellular and therefore rushes into the cell when voltage-gated sodium channels are opened. However, when voltage-gated sodium channels are blocked, the flow of sodium into the cell is cut off and it is impossible for threshold to be reached and for an action potential to fire from the axon hillock. Voltage-gated sodium channels can be inhibited both directly by the binding of Sigma-1 receptors and indirectly by Sigma-1 receptors through the activation of G proteins and protein kinase C. With these effects, Sigma-1 receptors can decrease sodium channel current as well as decrease sodium channel availability in order to decrease AP firing rate. However, there are multiple components contributing to the excitability of a neuron. Both potassium and calcium channels also influence internal neuron excitability. Sigma-1 receptors have been shown to inhibit voltage-gated potassium channels, which should depolarize the cell and create an excitatory response because the opening of voltage-gated potassium channels causes the cell membrane to become hyperpolarized and decreases the chance of an AP. Gated calcium channels are more complex than sodium and potassium because calcium is involved in the release of neurotransmitters and also acts as a second messenger molecule to trigger intracellular signaling pathways. Thus, the opening of gated calcium channels and movement of calcium into the cell is associated with both the release of neurotransmitters and intracellular signaling pathways. However, Sigma-1 receptors have been shown to both inhibit and facilitate the flow of calcium through gated calcium channels. With all of these voltage-gated ion channels being mediated at the same time by Sigma-1 receptors, the overall picture is very complex, and no studies have come up with a rule for the intrinsic excitability effects of Sigma-1 receptors. Instead, it has been shown that Sigma-1 receptors have different effects in neurons located in different sections of the brain. This is likely due to changing concentrations of voltage-gated channels as well as differential regulation of gated calcium channels. As noted earlier, Sigma-1 receptors can either inhibit or facilitate the flow of calcium through voltage-gated calcium channels. As shown, the influx of calcium causes the release of neurotransmitter. 2 main studies have been done on the effects of Sigma-1 receptors on gated calcium channels. The first showed that Sigma-1 receptors inhibit the flow of calcium in retinal ganglion cells. The second showed facilitation of the flow of calcium in the CA1 region of the hippocampus. However, few other studies have been done to test the effects of Sigma-1 receptors on calcium-gated channels. Many research articles have pointed to the Sigma-1 receptor as playing an important role in drug addiction, especially cocaine addiction. In order to gain an understanding for the Sigma-1 receptor's potential role in drug addiction, We must understand how Sigma-1 receptors act on gated calcium channels in reward centers in the brain, specifically in the nucleus accumbens. The nucleus accumbens, pictured above, is located lateral to the septum pellucidum and is involved in pleasure, reward, motivation, and addiction. In order to test for altered levels of calcium flow through voltage-gated channels upon Sigma-1 receptor activation, we must isolate and culture nucleus accumbens cells from neonatal rats in a physiological saline solution. Voltage-gated calcium channel currents as well as potassium and sodium currents can be measured under current clamp and voltage clamp methods described in a study by the University of South Florida College of Medicine. The cultured cells can then be tested while activated by various agonists and inhibited by antagonists as a control group. We expect that the use of Sigma-1 receptor agonists will cause an increase in flow through voltage-gated calcium channels and an increased neurotransmitter release in nucleus accumbens. Extensive research of Sigma-1 receptors is important because of the perceived role of Sigma-1 receptors in drug addiction, especially cocaine addiction. Current therapeutic approaches involve directly counteracting the blocking effects of cocaine on dopamine transporters. If, as this hypothesis suggests, Sigma-1 receptor translocation by stimulation with agonists causes facility of calcium flow, then cocaine can both block reuptake and increase flow of dopamine in the nucleus accumbens. This gives empirical evidence to the idea suggested in a recent study that the use of Sigma-1 receptor antagonists could have therapeutic value for curing cocaine and drug addiction and could serve as a breakthrough treatment for overcoming drug addiction.